Okay, and we are now recording, and this thing can see the board. Make sure anything I get on the board can be caught too. I didn't open Moodle to show you this, but let me tell you a couple of things I've done in Moodle to add for you. I put my schedule right up in bold at the top of Moodle, so you'll know where my office hours are and where my classes are. So if I'm not in my office, you'll know where I am. And my schedule, by the way, is always on my door, so you can always stop by and check there. If for some reason times I'm not in class or my designated office hours don't work for you, then you know what to do. You email me, stop by, let me know I need to talk to you, and we'll figure out a time. I'm generally here anyway. So if I'm sitting in my office, even if it's not office hours, I hope you stop in and say, hey. So that's how that works. I hope you'll help me out with that. All right. So, getting you guys. Today is Thursday, so this afternoon, 2:30, we are going to meet to start our lab times. Today, the purpose of our lab, according to our schedule, is to nail down what we're going to be doing for our project. Once that's accomplished, we'll take a little break, but we're going to come back together. We've got a 3-hour lab period, so we're going to work on this and hammer things out. Bring your laptops because you're going to need to get into the library and start looking some stuff up. And then we're going to get down to formulating what your research papers are going to be about and how we're going to partition out some work. Okay, so we're going to try to nail that down today in our lab time. So be sure to get your brain ready to work on that. All right? Okay, so getting back into your laptop. I've shown you by highlighting the process of this diagram. This might be, for example, a diagram you might see on your exam. Everything is there in diagrammatic form, and then you would simply write an essay explaining what's happening in that process. And some of the things we talked about there, dealing with, such as the notes that you might have made over here, dealing with the energy expense that costs the plant to reclaim that 3-carbon molecule to bring it back into the C3 pathway. So this is an expensive proposition. Other thing I mentioned to you was it's a useful proposition for some plants to have this ability to photorespire. It's believed, evolutionary plant biologists believe, that photorespiration is an evolutionary adaptation helping plants to adapt to high light, high heat environments, to be able to actually reclaim anything that might be lost during the photosynthetic process. So involving, as you can see, 3 different organelles in doing this. So the adaptive significance of photorespiration does benefit the plants, but it is also energy inefficient, energy expensive. So we believe that is why plants have been modified to be able to sidestep, ameliorate, make less expensive the process of photorespiration. Okay? Now, one thing I pointed out to you also in botany, every plant, all plants, big ALL, all plants do C3 pathway. The modifications come about in what to do with that enzyme that can interact both with oxygen or carbon dioxide. So rubisco is the problem, and I walked in here without my markers. It'll take a second after I finish my thought to go get them. Rubisco is the problem. And while it can interact with either oxygen, it has active sites on the enzyme that can interact with either oxygen or carbon dioxide. It is the CO2 plus rubisco interaction that is most beneficial to the plant. A straightforward reaction that gives you sugars once PGA or PGAL rather spins off from the cycle, okay? Right? But rubisco will interact with oxygen, so how do you stop it from doing that? How do you keep it happy with carbon dioxide? And that's where the modifications have come in. So little spooks there, because I'm sure you remember what's going on. Let me go ahead and arrow down here a little bit. Hey, down to C4 pathway. Oh, come on, in. Oh, okay. Okay. C4 pathway. Oh, too high. And take a look at the anatomy here. I want you to be see what's going on here. Remember that there are two different kinds of adaptations. One is more an anatomical adaptation, spatial anatomical, and the other is a timing or a temporal kind of adaptation. So C4 is our spatial. So take a look at this diagram real quick. I'm going to go get my markers and I'll be right back. Okay? I like to write on the board. Okay, let me make sure my marker works. Okay, so one of the things that differentiates a C4 plant from a C3 plant is anatomy. And it is a spatial distinction. Spaces become important. Regions or arenas of where things function. Enzyme location is what this is all about. What cell type is it in? Okay, and what is the enzyme type? So what we know is that the anatomy of C4 plants is something that has a bundle, a wreath of bundle sheet cells, or if you will, BSCs, bundle sheet cells. You might see me abbreviate that along the way. And for the C4 leaf anatomy, we've got a halo or a wreath of those cell types that surround the vascular tissue. And this wreath anatomy has led from spinning off from the original identifier of this distinction of anatomy. It's named after that person. It's called Krantz anatomy. Krantz anatomy, because I don't remember, I can never remember the name of the person. I'll have to look that up. But Krantz means wreath in German. And the original person who observed the anatomical differences was German. So that's one of the reasons that it got named that way. So Krantz anatomy, because of this wreath of bundle sheath cells. And what that does is it partitions out activities. So you can see here, here's the vascular tissue. We're looking at xylem and phloem, remember, as our vascular tissue. And adjacent to that vascular tissue is our BSCs, our bundle sheath cells. So this is just one lineup. Remember, this wreath is going to go all the way around that vascular tissue. So looking just here, we've got our bundle sheath cells, and then outside of that, the tissue surrounding the vascular tissue in the leaf anatomy is simply our parenchyma cells, right? Those tissues outside, they're called mesophyll cells in the leaf. Meso meaning middle, phyll meaning what? Leaf, right? So middle leaf area. Okay. So mesophyll to bundle sheath cells to vascular tissue, vascular bundle. all right? And compartmentalizing out where the enzymes are active is how we keep Rubisco from interacting with oxygen. And it's a shuttle system. We call it the PEP shuttle, PEP. Anybody taken biochem? What's PEP? Biochem 1 or Biochem 2?
I'm in Biochem 2 now.
Yeah. And did you cover photosynthesis in Biochem 1?
No, we're covering it now.
Oh, you're in 2? Okay. I didn't know where she dealt with that. Phosphoenol pyruvate. Phosphoenol pyruvate, PEP. If you've looked at cellular respiration and photosynthesis, PEP is in both of those systems. All living things do cellular respiration, right, of some kind or form, but only in autotrophic organisms like plants also do photosynthesis. So PEP becomes functional in both arenas, both the cellular respiration metabolic pathway as well as the photosynthetic pathway. So the PEP shuttle is simply, forgive me if I'm seeing a little, you know what that is? It's a taxi. And this little taxi picks up and lets off passengers. Guess who the passenger is? CO2. So literally, the PEP shuttle is a delivery system that shuttles carbon dioxide from where it's picked up out in the mesophyll cell, farther away from the vascular tissue, shuttles it in, taxis it in, and drops it off right in the bundle sheath cell. Well, guess what's going on in the bundle sheath cell? The C3 pathway. And guess who's there in the C3 pathway? Rubisco. But this thing is driven by something called PEP carboxylase. We've learned how to diagnose or just break out the names of things in biochem and other upper level classes. ASC tells us it's ASC ending tells us it's what? It's an enzyme. ASC ending tells us that it's an enzyme. Sorry, it's okay, we're getting on the same page here. So PEP carboxylase, carb, right? It's carbon dioxide carboxylase. So it's a shuttle for carbon, just it's in the nomenclature of the enzyme. Just kind of pointing out that we usually name things after what they do. I love that because it's not a mystery once you learn what the name is, you know. So PEP carboxylase, our little PEP taxi, our little PEP shuttle, picks up carbon dioxide out of the mesophyll, shuttles it into the bundle sheath cell, where it lets it off directly in line with Rubisco, keeping the concentration of carbon dioxide high inside the bundle sheath cell. So the bundle sheath cell containing Rubisco never reaches a point where oxygen becomes more prevalent than carbon dioxide, and Rubisco doesn't go fickle on you and go, well, look at there's oxygen, I'll go over here. Instead, look at all that carbon dioxide, I'm going to interact with that. So PEP shuttle C4 pathway is a modification, spatial, anatomical, about keeping carbon dioxide concentrations high immediately next to that enzyme that could go with oxygen if it was given the opportunity. Okay? Now the C4, this is the C4 pathway, and we are looking at, here's PEP carboxylase, and we have this 4-carbon molecule. PEP carboxylase brings in CO2, oxaloacetate, OAA. Oxaloacetate plus a carbon gives us malate, and that's our first 4-carbon molecule. Okay? Hence the C4 pathway. Remember, the Calvin cycle is C3. How does this get named C4? Because we bring in carbon dioxide, take our 3-carbon molecule PEP, which becomes the shuttle that brings it in. So oxaloacetate, PEP carboxylates, 3 carbons plus 1 carbon gives us 4 carbon. We bring that in and we drop off carbon dioxide. It goes back to a 3-carbon molecule, pyruvate. Recognize pyruvate from cellular respiration? So PEP and pyruvate, 2 things that are found in both metabolisms. And look here, there's an expenditure of energy that's needed in order for this to work. So you got to pay the tollman, right? So this is a little bit of an energy expenditure to do this, but it's less expensive than trying to reclaim photorespired, going through 3 organelles and figuring out how much energy or ATP is actually used in doing that. So a little bit of transport energy is needed, phosphorylation events, that kind of thing, to get this molecule to go back out to the bundle sheath, drop off our passenger, and then come back around and pick up the next one. You've got to pay the fare, a little bit of a fare paid there, okay? Now, what that means is we've got a 4-carbon molecule. We have a little bit of energy expenditure. There is a spatial distinction, and now there are 2 enzymes that are involved in this process. So I've just done the work for you. If you were listening, when I ask you to summarize what I just have told you in a bullet list of things, let's go for it. I kind of just did that for you. Let's pick it up and see whether you caught that. Summarize this for me. This is a spatial adaptation.
CO2 enters the mesophyll cells.
Okay.
And then transfer all the PEP shuttle to the bundle.
Okay. Okay. And what's the first 4-carbon molecule?
Malate.
What's the first 3-carbon molecule?
Pyruvate. Is that the
OAA, oxaloacetate. Mm-hmm.
Why does it say 4 carbon instead of 2 carbon?
Oh, I'm sorry. It's an exchange there. Beg your pardon. I can't read very well on here, and my cursor's back over here. You are absolutely right. OAA is your first 4-carbon molecule. I almost said that, but then I couldn't see the 4 next to it, and I figured I was wrong. But I was right after all. Okay, so OAA is a 4-carbon molecule, and it is PEP, which is our 3-carbon molecule. PEP plus CO2 gives us oxaloacetate. That's our initial product. Beg your pardon. I knew that was right, but it didn't look like it on the board. Sorry about that. Never know whether to trust your own instincts or what you see on the board. You're exactly right. Now, it exchanges into a different form of the 4-carbon molecule, malate, so that it can actually be brought into the bundle sheath cell. And I started to say that, and I was like, wait a minute, that doesn't look right. The cursor was sitting over that note. Okay. Have I exonerated myself in that? Sorry about that. But you're absolutely right. Okay, so our first 4-carbon molecule is OAA. It's transformed into malate so that it can be transported from the mesophyll cell into the bundle sheath cell. It has to come in that form. There's a transporter there that allows it to pass through. And when that comes in, it drops off your CO2. CO2 gets shuttled in, and this is where rubisco is. And it picks up the carbon and goes through the process of C3 pathway, just like we talked about last time we were together, way up at the top, okay? Light-dependent, light-independent reactions. Okay, and then we make sugar, and that sugar then gets transported through the phloem inside the tissues of the plant. Phloem is where we see food moved. Xylem is where we see water moved, right? And sugar is food for the plant. So this is our spatial anatomical difference. Okay, so just to kind of bullet list, we started out back correcting ourselves. So this is a spatial anatomical distinction. It has a C3, C4 molecule distinction. C3 is PGA, C4 is OAA initially. Okay. It has Rubisco dealing with the PGA. It has PEPCO, carboxylase, PEPCO is a way of abbreviating that for making OAA. So in the C4 pathway, we have two enzymes working now. Make sense so far? Summarizing what we've seen so far? Okay. And basically we call this a shuttle system. And we call it the PEP shuttle. That transports carbon from the mesophyll cell into the bundle sheath cell, where all the regular stuff for photosynthesis happens without Rubisco going wild on oxygen. Anything else that you could summarize or bullet list or kind of itemize as things that we've seen? And you could go off a little bit on, well, it's spatial anatomical. That means that there's mesophyll cell, bundle sheath cell, vascular bundle, okay? And that the wreath of bundle sheath cells is called krantz or wreath, and that's why we call it Krantz anatomy. Getting how you pick up all the stuff you need to let sink in, think about, and then give back to me on an exam in an essay format. Making sense? Pointing it out as we go along earlier, and then hopefully you'll get the hang of it, and it becomes a little bit easier for you just to say, I see what she's going to want us to say there, okay? Let me see here. I think that pretty much, and I should get some kind of award for my taxi drawing on there. See the little taxi sign up on top there on the roof? I've only taken a taxi one time in my entire life. How about you guys? No? And you have? Where?
In New York.
New York taxi? Really? Ooh, that was an experience, I'm sure. Yeah? Taxi?
Yeah.
Have you?
Same place.
Really? New York City? Yeah? Were they kind of like what everybody thinks about with the New York City
Scary.
taxi?
Yes.
Oh gosh.
I wouldn't want to drive up there at all.
You'd rather take the subway, right? Something like that. No, not even that,
Subways are
huh?
scary too.
Yeah, subway's really sketchy.
Yeah.
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 Yay. Loves New York City. I've never been, truthfully, I've never been to New York City, but I've been to either San Francisco, Chicago, but I've never been to NYC. Isn't that terrible? I need to go. I really do. Taxi? Yeah? Where?
Places in Florida.
Yeah?
Should be different.
Cool. So not quite as terrorizing as New York
No.
City, but they're probably a lot nicer in cities like that. Definitely. I took my one and only taxi in Caracas, Venezuela. Yeah. And that was very interesting because I obviously couldn't speak the language very well, and they just were like trying to ask me, where do you want to go? And I'm like,
I've taken one in Nicaragua, and there they don't have like road rules like we do, and it's crazy.
Exactly. Yeah, they're scary. It's really scary. In addition to a taxi, though, I don't know if this counts, but when I was staying in Merida in Venezuela, you got around by transport vans, basically, like a 9-seater van. And it's a 9-seater. There would be 24 people. on that van. And they're literally, I mean, they're literally, the door's open and they're hanging on and they're hanging out in the van as you go from A to B, you know? And they hop on and they hop off. And it's just like, really? That's the one that I want to take, huh? Very strange way of doing things. But adventures, good stuff. You can tell stories about it. Okay, everybody okay with this one? We're going to go on and look at the CAM pathway, which has been such a special discovery because it is actually a pathway that is unique to certain types of plants, CAM plants. We actually call them CAM plants. CAM. Now, did you guys get PEP was phosphoenol pyruvate? You got that down? CAM is crassulacean acid metabolism. Want me to write it down? Okay, crassulacean, CAM is crassulacean acid metabolism. And I'm going to go get me another marker. I cannot stand not being able to see what I'm writing on the board. Crassulacean acid metabolism, and there is a family of plants called the crassulacean family, Crassulaceae, family ending, okay? And those plants in that family are have a higher incidence of doing CAM photosynthesis. The types of plants that you find that do C4 pathway are corn, wheat, rye, barley, grasses in general. Grasses in general are the kinds of plants that are typical in hot, windy, dry environments, right? You find the wheat belt where? Nebraska, Kansas, right? Arkansas, Oklahoma, the plains, the central plains of the United States, and that's where we see those things so well adapted to live. So largely speaking, it's the Poaceae family or the grass family that's C4, but there are other examples, some broadleaf plants that are also C4. But the CAM pathway, crassulacean acid metabolism, is pretty much reserved for succulents and desert types of plants. Okay, let me get another marker. And then what you're going to do after we go through all this, our little activity is down here. And we're going to go to your textbook and start to look through for rates, efficiency, and optimism in comparing C3, C4, and CAM. Okay? I'm going to get there in just a second. I'm going to get a marker, go through CAM with you. Yeah.
No, she's talking. It's not her talking.
Oh, yeah.
Yes, she's talking to Eric. I heard her go. Well, it might be a little bit longer. She wants to guess. Why did you just class? Because it's one. No. Oh, well then I don't know why you did that.
It's just a bio elective.
It wasn't supposed to be this.
It was assist, but
It wouldn't have been nearly as bad, though. You know,
Teach 'em more.
conservation like
yeah.
that class was great.
The test was very. But this. What, did you see this in the first back? It wasn't even in this place. Yeah.
Yeah, that was easy for me. Well, I took it the year before y'all did, so we had like a test every week. It wasn't bad at all. He failed tests and still we found the pass.
Yes, we had, what, like 2 tests?
Yeah, we had
Two,
like
maybe
2 or
3
3
tests.
And we had 3 and everyone bombed the third one. I remember that. Like, nobody passed it.
Hey, I passed every test in my class, and I got A's too.
Yes.
Girl, then you were like the only one.
I was the
Because
she ended up giving everything. She ended up giving me.
Yeah, most people have lived in like a third.
Yeah.
She screwed up everything like that, like most people were expecting.
Yeah, no,
It's like worse than organic.
Yeah.
But then he doesn't care half of them, so. I'm happy I never have to take a class from Piper though.
People be dropped environmental. Why
Oh my
do you do that?
God.
You don't have to do. You don't have to make it happen.
Because I needed the hours, because they wouldn't add my independent study, so I had to have the hours on there and then they added the
What's your independent study in?
evolutionary.
Evolutionary biology.
Oh, yeah. But I mean, semi-electric biochem.
Beg your pardon, sorry about that.
Yeah.
No, just biochem one.
So you're staying next year semester?
Okay, you're fine, okay. All right, so we talked about how how C4 pathway is a spatial adaptation. CAM is a temporal adaptation. So CAM is a time issue. And it's literally night and day. Literally night and day. That's what we're talking about. As you can imagine, the environments that we actually see, succulent plants, desert plants, it's extremely hot during the heat of the day. And if they opened their stomates, then to let carbon dioxide in that they need as a resource in order to do photosynthesis, if they open them up, then of course water gets out. And that can be very dangerous for desert plants. So what they do is they open their stomates at night when it's cooler and the diffusion evaporation factor is much more reduced, and they bring in carbon dioxide and store it into a 4-carbon molecule. And then during the daytime, where we have the light-dependent reactions happening as well as the light-independent reactions happening, then carbon dioxide is released. It's been stored, sequestered away, and then they release carbon dioxide during the daytime so that they can function to make sugars, okay? Now, that's the short end of it. But what I want you to notice is that a lot of this is about trade-offs. The trade-off is open the stomate, you need CO2 to come in, but water always gets out at the same time. And as you remember, we need to split water to release hydrogen, electrons, and oxygen, right? We need to split water. If you start losing water, the plant gets dehydrated, can't do photosynthesis, and frankly, no other metabolism when it's that stressed, and it just won't function very well, okay? So these guys, these adaptations, C4 and CAM, are trade-off adaptations to minimize water loss while maximizing uptake of CO2, okay? So big thing about these two things is that they are trade-offs. Now we look at the structure in the CAM pathway, and we have, let me get myself oriented here. Here's our stomate, our opening to the world where we have atmospheric carbon dioxide coming in. And let's see, stomate open, stomate closed is the difference between these two. Dark means, guess what that might mean? Dark means night, light means day. So this is what's happening. Let's start over here, because this is what happened. No, I'm sorry, start here. This is what happens when we bring carbon dioxide. And note we have here, look here, PEP carboxylase. So again, we're using that same enzyme in the CAM group of organisms, plants, in order to function in storing and sequestering away carbon dioxide. So we use PEP carboxylate. We bring in atmospheric carbon dioxide, PEP carboxylate. First molecule is OAA, or oxaloacetic acid, okay? And then we go through some, we deliver a little bit of energy through NAD becoming NAD, NADH becoming NADH plus. We've handed some hydrogens and we've handed some electrons to this 3-carbon molecule. Here's the enzyme that's important in that process, NAD malic dehydrogenase. And we go from oxaloacetate 4-carbon molecule to malate 4-carbon molecule. And it goes into a little holding pond. It's a malic acid vacuole, that membrane-bound structure that exists unique to plants, not found in animal cells. Vacuoles. Remember them? Okay. So we store that there. And notice the proximity within the cell of the plant that is close to the chloroplast where the C3 pathway is cooking away, right? Inside the C3 pathway, we've got the light-dependent reactions, and we have starch being broken down to triose phosphate, which becomes phosphoenolpyruvate, or what's that? PEP, and pick up carbon, 3 carbons plus carbon becomes 4 carbons, energize it, shift it around a little bit, becomes malate, malate gets stored, malate releases carbon, it may get moved into storage of a starch molecule, right? So over here, and it can be broken, it can just be brought in as CO2, it could also be brought in as starch, stored there, broken down, CO2 gets released, but the starch then can energize triose phosphate, which then becomes phosphoenolpyruvate, which picks up another carbon, creating another 3-carbon molecule. Okay, all of this is happening to create a storage area of 4 carbons, and that 4 carbons is going to release one of the carbons. So take it over to the daytime. It's too hot outside, we close our stomates, okay? And here's our malic acid in the vacuole, here's our malic acid, and here's our chloroplast, right, in proximity here. So this is just about cooking the books, making more and more malate and storing it away at nighttime. Over here, we take malic acid, export malate, and use the NADP plus malic enzyme and release the carbon dioxide. So we're cutting carbon off the 4 carbon. This becomes 3 carbon, right, and goes back through the process. So malate releases the carbon dioxide. It becomes, it goes into the Calvin cycle and does the C3 pathway. Malate 4 carbon, release the carbon to pyruvate, pyruvate to starch, can be stored back to starch, or it can also just go back through the process again. Starch becomes a resource for this storage process. So desert plants have a couple of mechanisms going on. They hold on to sugars in the form of a storage molecule, starch, right? Starch is a carbohydrate, which is a very long structural molecule that's sugar, 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 right? C6H12O6, C6H12O6, it's all starch. So we're storing carbon in that way, and we're also storing our 4 carbon molecules in malic acid vacuole, little holding pond. Okay, this happens. CO2 is released, so the light reactions in the chloroplast can go along and make sugars stored in starch. This happens so that we can open the stomates, let carbon dioxide in, and create a pool of carbon that can be used during the daytime hours. Pretty simple. Questions? C3, C4, and CAM. And I want you to use your textbook, work through some comparisons. The intention here was for you not to cheat and look ahead. I want you to be able to use your textbook in order to glean some information that I'm asking you to work with. So give it a shot. Let's see you guys work together on getting some information down on there, and we'll check you. We'll look at my completed table below. So if you will, you'll have to use my book. And take, let's say 10 minutes, okay? It'll be noon in 10 minutes, just to see how far along you get. And this, again, is the second week of classes, so we'll see, get you guys used to doing this, okay? So you're comparing rates, you're comparing efficiencies, and you're comparing optimums. And see how your textbook unfolds that information for you. And I think, Luke, I can pause you because we've just gone over this diagram in your notes. And they are now working on this, as you can see, and then this is my work in gleaning some notes that we're going to take a look at in just a couple of minutes. So I want to ask you also to do the same thing. See where you can find this information in your textbook. You can work backwards if you want on your own, but I want to see this table filled in by hand when I see you. So you'll have some information. I'm going to pause this recording until we come back and go over my completed table. So see you in a minute. Back. So I've just been giving a little diatribe about one of the reasons that we're filling in this comparative table. And it takes students who are working through learning a little bit further along when you're actually working to make direct comparisons. So I am going to take you down to this table that I completed for you. Let's take a look at it together. I want to be sure that you are comfortable with having perhaps found this for yourself in your chapter reading. So when you're taking chapter study notes, this is the kind of thing you might actually do as you're completing your chapter study notes. Okay? So let's take a look. We've got some C3 notes, C4 notes, and CAM notes. Here's our rates, efficiencies, and photorespiration disadvantage, and I think it goes down to advantages and then optimums down below. I'm keeping the font big. We're just going to make some across the table comparisons. Let's look at this together. So what we've learned about C3 and what we've learned about C4 and CAM. It's powered by Rubisco. It has a low affinity for CO2. It's most limited in warm, bright areas, which is what we talked about, why grasses do so well in those areas, because they're C4. There is 10 to 30% total nitrogen in leaves. It's bound up in Rubisco. CO2 in the atmosphere, if we have high concentrations of CO2, we have higher photosynthetic rates. Okay. What our knowledge on that point has actually generated a lot of research. Scientists are actually studying greenhouse situations where they can pump in or amend CO2 concentrations in a greenhouse atmosphere. How does that affect CO2 rates for certain types of plants? You can create little tents over species of plants, increase CO2 in that area, and see what it does for photosynthetic rates here where you're adding CO2 and over here where you're not adding CO2. So you can do a little bit of an investigation. Well, what if it does so well with CO2, what if we give it more CO2? And hey, this global warming thing with CO2 in the atmosphere increasing, shouldn't our plants be just taken off? Well, in some cases they are, but you know, in other cases that's not a good thing, depending on the plant. Okay, C4 notes, C3 still converts CO2 carbs. See that? All plants do C3. See that? Initial fixation from the atmosphere reduces photorespiration, loss of energy, and CO2 respired. Photorespiration over here is a problem. It's a disadvantage. PR is photorespiration. And the first column down here is a disadvantage. See that? So an additional step, and it costs some ATP, right? But it recuperates losses that are expended, that are more energy expendable over here in C3 plants. CAM notes are C3 is still converted to CO2 carbs, essentially the same biochemical pathway as the C4. You get a C3 PEP, add a carbon, becomes OAA. OAA to malate, we saw that, right? And then malate in the CAM gets stored, or malate gets shuttled into from the mesophyll to the bundle sheath cell, where it drops off that carbon dioxide. Okay, so distinct differences, but definitely very similar to the way they do things between C4 and CAM. The C4 adaptations to this Pepco has a high affinity for CO2, where we already know that this is a low, Rubisco has a low affinity for CO2. It's found only in the mesophyll. It's a spatial adaptation and anatomical adaptation, keeping oxygen and CO2 at the site, oxygen CO2 ratios at the site of Rubisco fixation. You want oxygen to be low, CO2 high. CO2 internally liberated by decarboxylation of OAA or malate, depending on the enzyme involved. Over here, the adaptations, we have Pepco, again, high affinity for CO2, Pepco in all photosynthetic cells. Temporal, spatial, temporal separation of oxygen, carbon dioxide, where Rubisco is fixing CO2. CO2 internally is liberated by decarboxylation of HEP here, and decarboxylation of OAA or malate here. See the difference? Direct comparisons, okay? And moving on down. C3 rates. They're not exactly lined up, I apologize for that. But for photosynthesis, it's a major impact. Concentrations of CO2 definitely limit the rate of photosynthesis. And if you look down here to C4 rates, higher maximum photosynthetic rates for C4. And if you look over here, CAM rates, day-night separation slows maximum photosynthetic rates, reduces accumulation of carbon versus the C3 or C4 CAM plants, rather. Okay, so direct comparison of rates between all three of those things. And then optimum. C3 has a lower temperature, optimal temperature, lower optimal temperature for photosynthesis. It has a higher need, for a higher concentration of CO2, and light saturation at levels well below full sunlight. So it can work well in shade. It's shade adaptive. And remember when we come back, come back up here, it says good in shade. See that? Okay. So we've got optimums for C3, light saturation levels well below full sunlight, and that translates to efficiencies being good in shade. See that? Okay. Meanwhile, back over here, one more time, we've got rates, higher maximum photosynthetic rate than C3, and optimums much higher than C3 do not saturate even in full sunlight because carbon dioxide uptake is not limited by high oxygenase activity of Rubisco. Over here, optimums good even in warm temperatures, so deserts, hot, windy, dry for C4. See how well they do and reasons why they do better than what we see C3 doing all by itself. All right. You've got this in your, if this is posted up in Moodle, you've got this to look at. I want this to be an object lesson. You too could have found this information. I did exactly what I'm telling you to do. I read through the chapter, I gleaned everything and made bullet list of comparisons, went back and just said, oh, look at that. There's rate comparisons, there's optimal comparisons, and then I just created myself a little table, okay? And I want you to get familiar with doing those kinds of things as you go through. And there's more down below, C3, C4 CAM, looking through all of that. Okay, it's going to take us into photosynthesis, looking at it through an evolutionary timeline. And we'll pick up there. I'm going to try to get us through chapter 2 on Thursday or Tuesday of next week, okay, to try to wrap up and get us kind of on schedule. This afternoon, we have some stuff we talked about on Wednesday that you might be interested in, because these guys meet with me a little bit on Wednesday. You all are invited to do that at 10 o'clock on Wednesdays if you want to join us in my office. But meanwhile, we're going to get our heads together and talk about our projects. Okay? See you guys 2:30, yes? In here. See you guys at 2:30. Okay.